Okay, for those of you watching this recording, this is not the live workshop session. We had some technical difficulties trying to record that, so I will put together a separate recording of these slides just for continuity, for you to refer back to if you were there and want to check on some things. This will probably be a little bit more brief, a little bit less detailed than the actual session, but should have all the essential information. If there's anything that's not clear, please just let me know by sending a message in Slack or otherwise. All right, so let's get into what we're going to cover today, which is photoluminescence quenching. So let's start with some definitions off the bat. Simply put, photoluminescence quenching is any process that reduces the quantum yield and/or lifetime of a photoluminescent process. And usually both the quantum yield and the lifetime are reduced when this occurs, as we'll talk about in more detail later on when we get into the mathematical relationships that are involved in quenching. And as we'll also see, there's a large variety of different processes that can lead to this. So they can be intramolecular, they can be bimolecular, there's different mechanisms and things at play. So we'll get into some of that. We're going to touch, again, not on a large level of detail, but kind of scratch the surface of this type of process. All right, so the first major category of photoluminescence quenching is energy transfer, which will be abbreviated as ENT to distinguish it from electron transfer, which is just abbreviated as ET. So in energy transfer process, there is no net transport of charge. And so as we'll see, there's a couple different ways that this can happen. But essentially what's occurring is that your excited state molecule, so labeled here as D star, and that's going to then interact with an acceptor molecule A, and it's going to transfer its excited state energy to the acceptor. So again, the notation we use here is donor acceptor. Typically, donor is the molecule that is initially excited and then transfers energy to the acceptor. So there's no net transfer of charge. If D and A are neutral molecules at the start, they are also neutral molecules on the product side. But this process involves the transfer of excited state energy. So the donor molecule would normally luminesce in the absence of the acceptor, but because it's transferring excited state energy to the acceptor, the amount of luminescence that you get from the donor reduces in the presence of the acceptor. Now, the acceptor may or may not be photoluminescent, so when you generate the excited state of your acceptor, you may see photoluminescence that arises from that excited state, or if it's not luminescent, you won't observe it directly, but this excited state would still be populated. All right, so within energy transfer, there are two categories. And the first one here is Förster resonance energy transfer, abbreviated as FRET, and often referred to in that way. It's not correct to call it fluorescence resonance energy transfer. A lot of the biology literature and even some chemistry literature refers to it as such. It does often involve fluorescent molecules, so it's not wrong from that standpoint, but the correct way to denote FRET is as Förster resonance energy transfer, named after presumably the person who discovered it. All right, so let's get into what it involves. So it involves emission and absorption of a virtual photon. This has a very quantum mechanical origin to it, but so we're, again, not going to talk about it in that level of detail. But what essentially is occurring is shown here in this Jablonski diagram. All right, so you excite the donor molecule via excitation. Vibrational relaxation populates the lowest level of S1. Now, from this S1 excited state, in the absence of an acceptor, fluorescence can occur. That's shown with the green arrow here. But the FRET process, shown with this orange rectangle, occurs when the acceptor is in the vicinity, or it can occur. So what's essentially happening is you don't actually emit the photon and reabsorb it, but more or less in net the process is the same. So a virtual photon is emitted by the donor and reabsorbed by the acceptor to generate an excited state within the acceptor, usually, again, the S1 state following vibrational relaxation. So there's no actual emission or absorption of a photon. I want to make that part clear, but it's kind of like that process is occurring. It's almost like you're emitting a photon from S1 to the donor, and that photon is being immediately recaptured by the acceptor, but you can't observe that photon from the donor. It's a virtual photon, as I called it up here. Nonetheless, because this process can almost be thought of as emission and reabsorption of the same photon, it requires that there be overlap between the fluorescence of the donor and the absorption of the acceptor. So this fluorescence needs to occur at a wavelength where absorption can occur within the acceptor. Okay, so that's how FRET would occur. And there's also a steep distance dependence in FRET, is 1 over r to the 6. So as the donor and the acceptor separate in distance, where r is the distance between them, the efficiency of FRET, the percentage of times that it would then occur, falls off dramatically. So this dramatic distance dependence is one of the hallmarks of FRET, and as we'll talk about, is one of the ways of distinguishing it from the other type of energy transfer that we'll discuss. All right, so that's going to be FRET, Förster resonance energy transfer, and again, it's this virtual photon being emitted and absorbed from the donor to the acceptor. The other major type of energy transfer is called Dexter, and for this I have a video that I'll show if it'll let me play. Here it is. So Dexter energy transfer process, as you'll see here, is what's called a double exchange process. So in Dexter energy transfer, let's see what occurs in this case. It's simplified with sort of a HOMO-LUMO type diagram here. So the donor molecule is going to be excited eventually, so you'll see the electron being promoted to the LUMO, and then from there you're going to have 2 simultaneous electron transfer events happening. So if you saw what happened, basically the electron from the LUMO of the donor transferred to the LUMO of the acceptor, while at the same time an electron from the HOMO of the acceptor transferred to the HOMO of the donor. So you have electron transfer going in both directions. So in net there is no net electron transfer. It's just energy transfer where you're generating now an excited state of the acceptor, but not any sort of redox product. The distance dependence for Dexter energy transfer is different. It's proportional to e to the minus r, where r again is the distance between them. So it's not nearly as steep of a dependence as FRET, and so that distance dependence, which also then manifests as a concentration dependence if you're doing it bimolecularly, that can be a way of distinguishing FRET from Dexter. And another thing about Dexter is that it may involve either single states or triplet states. It's typically the spin is conserved. So if your donor involves a singlet state, as we showed in the video, it will produce a singlet state in the acceptor. But if the donor is able to populate its triplet state, you can then do the same type of double exchange process and get to the triplet state of the acceptor. So it can involve both. In contrast, FRET is almost always going to be just 2 singlet states involved, because it involves that virtual absorption process, which is not going to be very probable or very likely to occur if it's a singlet to triplet transition. So FRET is almost always singlet to singlet, but Dexter can be both, and very often in the context of our research we do see this type of triplet energy transfer, which most likely occurs via Dexter in all these cases. All right, now a process that's related to these energy transfer pathways that we've discussed is called photon upconversion. So this is really just an extension of mostly Dexter energy transfer, I suppose, although it could involve FRET as well. But it's a more complicated process, and one that has important applications in photonics and photovoltaics and things like that. So we'll get into applications at the very end of this, but it's an important process. And what it does is it converts 2 low energy photons into a high energy photon. And so there's some energetic requirements here, which we'll go through in a little bit after I show the diagram. So basically the first step of photon upconversion is triplet energy transfer, occurring in the exact same way as I just showed you. This time I'm just showing it in terms of a Jablonski diagram, but again, what occurs is that your donor is excited. This requires triplet states, so you're going to cross over into the T1 state of your donor, and then from there, triplet energy transfer populates the T1 state of the acceptor. And so this process has to occur twice in order for photon upconversion to occur. So this same process occurs 2 times. In order for this process to occur, the triplet state of your donor needs to be higher in energy than the triplet state of the acceptor so that the energy transfer is favorable. And then once this occurs 2 times, you generate 2 acceptor triplet states, and 2 of those triplet states come together and do what's called triplet fusion. So in triplet fusion, 2 T1 states interact with each other, and that generates an S1 state while the other one simultaneously relaxes to S0. So in order for this to occur, this S1 state needs to be less than 2 times the triplet state. Basically, the sum of these two identical triplet states needs to be greater than the S1 energy in order for this process to be energetically favorable. But it generates a singlet excited state and a singlet ground state, but from that singlet excited state, you can then get higher energy fluorescence. So it's possible in this process to use 2 low energy photons to excite the donor, and then at the end of all of this, higher energy photons are emitted from the acceptor. So that's why we call it photon upconversion. The maximum quantum yield is 0.5 because in order for 1 photon to be produced via fluorescence, at minimum 2 photons need to be absorbed by the donor. So 1 emitted photon for every 2 absorbed photons means that your maximum quantum yield is 0.5. So that's just another related process that is going to involve these types of energy transfer pathways. All right, and then the electron-transfer quenching, ET as it's abbreviated, that process is going to involve now net redox chemistry. So you're going to have, in this case, charge transfer between the donor and the acceptor molecule after excitation. So you're going to be doing just a single electron transfer event. So in this case, in the energy transfer, we always said the luminescent molecule was the donor. It was donating excited state energy to the acceptor. But conventionally, the donor-acceptor notation for redox chemistry, the donor is the molecule that that gives up the electron. The acceptor is a molecule that takes in the electron during the redox process. And so in electron transfer quenching, the luminescent molecule, the molecule that's excited initially, could serve as either the donor or the acceptor. And this electron transfer quenching can also involve singlet or triplet excited states, but triplet states are very often involved because these electron transfer events are bimolecular in nature. They require the excited state molecule to collide with the donor or the acceptor that it's reacting with. And because it requires that diffusion and that bimolecular reaction, you need to have a relatively long-lived state, and triplet states are typically longer-lived. All right, so if the excited state molecule is serving as the donor, we call that oxidative quenching. So in oxidative quenching, there's a little bit of a typo here. There should not be a star there. That should just be a n-1. So my laser pointer is covering up the little asterisk that shouldn't be there. But in oxidative quenching, you excite the donor, and from this excited state, it's going to transfer an electron to the acceptor. So that oxidizes the donor, reduces the acceptor, you get charge separation, and you generate 2 ground state molecules, one oxidized, one reduced. And we call it oxidative quenching because the donor molecule, the excited state molecule, is being oxidized in the process. So a simplified orbital diagram of how this might look, this requires that the LUMO of the donor molecule after excitation is higher in energy than the LUMO of the acceptor. That allows an electron to be transferred from this LUMO of the donor to the LUMO of the acceptor in a downhill process. So it's a single exchange in this case. Dexter energy transfer was double exchange, but this is a single exchange process where one electron is being transferred, and that results in, again, formation of the oxidized donor missing 1 electron and the reduced acceptor, which now has 1 extra electron up in its LUMO. The other process that can occur, which is also a form of electron transfer quenching, is reductive quenching, and in this case, the excited state molecule serves as the acceptor. It takes an electron from the donor that it is reacting with. So again, the net process is transfer of 1 electron to form a reduced and oxidized species, but this time the excited molecule is the one that's accepting the electron. And in terms of the orbital diagram, what happens in this case is you excite the acceptor, which really results in 1 empty position in the HOMO, 1 unoccupied HOMO spot, and then an electron from the donor is going to transition into that. So it's a single exchange process, which then generates the oxidized donor and the reduced acceptor, but this time it was the acceptor that was initially excited via photoexcitation. All right, so that's going to be the process of electron transfer quenching, both oxidative and reductive quenching. The other part of this that's important is going to be the excited state redox potentials, which dictate the thermodynamics of this process. Because as we said, in order for these energy or electron transfer processes to occur, they need to be thermodynamically favorable. You're not going to do, just like in ground state chemistry, you're not going to do thermodynamically uphill processes very often. But in excited state redox chemistry, the way that that is sort of determined is via the excited state reduction potential or oxidation potential of the molecule that you're photoexciting. So when your molecule is serving as a photoreductant, meaning that it would be oxidatively quenched, it's donating electron to the acceptor, the way that you estimate the relevant potential for that, which is shown here, is two terms. The ground state potential, which you measure by some sort of electrochemical technique, usually cyclic voltammetry or sometimes differential pulse voltammetry. There's other ways of doing it, but you use electrochemistry to measure a ground state potential. And then you measure the E00 value, as we talked about in the last couple lectures. So E00 is the energy gap between the 0th vibrational levels of the excited state and the ground state. It's the maximum amount of excited state energy that you have available to you during this type of quenching process. And you can estimate it in a few different ways using photoluminescent measurements. So you can either look at the crossover point between absorption and emission, as we talked about last time, or if you do a low temperature photoluminescence measurement and you can resolve the vibronic structure very well, typically the first vibronic peak would be the E00 value. So those two terms together, ground state potential, E00, give you an estimate of the excited state reduction potential for a photoreductant, and the significance of this value is that the more negative it is, the stronger the photoreductant you have. And in order for oxidative quenching to occur, you would need this excited state potential to be more negative than the reduction potential of the acceptor that you're transferring the electron to. Then for photooxidants, pretty much everything I said is just true, but in the opposite order. So for a photooxidant, again, to estimate the potential from the excited state, you need a ground state potential, this time for reduction of the acceptor, and the E00 value, which you get from photoluminescence. Those two added together gives you an estimate of the excited state potential associated with transferring an electron to the excited state of the molecule you're studying. And in this case, for a photooxidant, the more positive this value is, the stronger a photooxidant it would be, and this potential needs to be more positive than the potential of the donor, the oxidation potential of the donor, for that electron transfer process to occur thermodynamically favorable. Now what we're going to finish with for the rest of today's abbreviated recorded lecture. Again, I'm going through this a little bit faster than I did during class. If you have questions or want further elaboration, please get in touch with me. But we're going to finish with a description of quenching kinetics. All right, and so this is sort of what we would measure experimentally when quenching is occurring. And whether we have energy transfer or electron transfer, whether it's Dexter or FRET, whether it's oxidative quenching, reductive quenching, the quenching kinetics behave identically in all those cases. So they can be measured the same way, they can be evaluated the same way. Now the sort of other side of the coin then is that because the quenching kinetics are the same no matter what type of quenching process, you can't really use the kinetics to tell you what kind of quenching is occurring. So if you want to know whether it's electron transfer, energy transfer, or what type of electron and energy transfer processes, additional analysis is needed beyond just measuring the kinetics. But the kinetics are still very useful to measure, and they're very straightforward to measure using photoluminescence techniques. And this is being covered here because understanding how these kinetics are measured does require knowledge of photoluminescence measurements that we covered in the last workshop session. So that's why it's sort of in this sequence of the course. All right, so to start with, let's look at a simplified Jablonski diagram that includes now a quenching term. So in the absence of a quencher, so when we talk about quenching kinetics, we're going to use Q to abbreviate what we call our quencher molecule. That's going to be the molecule that's either participating in energy transfer or electron transfer with the excited state. So it can be, it can be a donor or an acceptor, as we talked about, and we'll just use the generic abbreviation Q for whatever molecule is reacting with the excited state molecule. So in the absence of the quencher, with no Q present, you would have this simplified Jablonski diagram where you have fast excitation, as we talked about last time, and then radiative and non-radiative decay competing with each other to deactivate the excited state back to the ground state. When you introduce a quencher, you introduce a new non-radiative decay pathway. So it's shown as a squiggly line here because it's non-radiative whenever you have quenching. But the rate of that process, the rate constant for that, is a product of the inherent quenching rate constant KQ and the concentration of the quencher, because these are, as we're talking about, is a bimolecular quenching event, so it depends on the concentration of the quencher molecule that's present. So this new non-radiative decay pathway is bimolecular, and that then modifies the lifetime of the quantum yield. So again, in the absence of quencher, the lifetime is just the inverse of Kr plus Knr. But now when you have a quenching process, you add in this new term here, this bimolecular term, and that reduces your lifetime tau. The amount that it reduces is dependent on the concentration of the quencher. And similarly, the quantum yield, which is Kr divided by Kr plus Knr, as we covered in the last 2 lectures, when you introduce a quenching process, that term goes in the denominator, and that likewise reduces the quantum yield, depending on how much quencher you have. So typically quenching processes will reduce both the lifetime and the quantum yield. We'll talk about exceptions to that in terms of in a little bit, but in general, this is what you'll see when you have a bimolecular quenching process. So the way that we measure the rate constant KQ, all right, so the rate constant KQ is going to let me go backward. Doesn't let me go backwards. So KQ, which is the rate constant for quenching, is measured using what's called Stern-Volmer analysis. So what you do in this is you basically measure the photoluminescence intensity or quantum yield. Technically it's the quantum yield, but you can do it with intensity as long as you keep the instrument settings identical through the whole measurement. So that, remember we talked about last time that the observed intensity depends on a lot of factors. As long as you control those factors, you don't necessarily need to measure quantum yield every time. You can just do it using the integrated or peak intensity. And then you can also measure the lifetime. And you do those measurements as you increase the concentration of the quencher molecule. And so basically rearranging the equations from the last slide a little bit, I'm not going to go through the derivation, but you derive what's called a Stern-Volmer relationship, where these ratios, so phi 0 and tau 0, those are the quantum yield in the lifetime in the absence of quencher, the native quantum yield in the lifetime of the molecule. Phi q and tau q are the quantum yields in the lifetimes in the presence of a certain amount of quencher, and those are going to be reduced when the quenching process is occurring. So that ratio is going to have a simple linear relationship with an intercept of 1 and a slope here that we call the Stern-Volmer constant, KSV. And the Stern-Volmer constant is a product of 2 elementary terms, tau 0, which is the lifetime, the native lifetime of the excited state molecule, and KQ, which is that quenching rate constant that we're often interested in measuring. All right, so KQ is going to be the quenching rate constant tau, and we get those from this Stern-Volmer analysis. So you measure the photoluminescence intensity and lifetime with increasing quencher concentration, and this plot gives you the relevant parameters. Now there's some other considerations. So ideally, your quencher molecule Q should not absorb at the excitation wavelength that you're using, because that will filter out some of the excitation wavelength and reduce the observed intensity of the photoluminescence. Now, it's sometimes not possible to avoid that situation, so if the quencher molecule does absorb at whatever wavelength you're exciting, there's some math you can do to correct for that, and you'll just correct the observed intensity by that amount of reduction in the photon flux. I'm not going to go through that math here, but if it does come up in your research, make sure you're accounting for that, and we can talk about how that works if it does become a consideration that you need to worry about. So you have to, and you also have to avoid completely filtering out the excitation lag, because often you use a large excess of quencher. So if your quencher absorbs really strongly at the excitation wavelength, if you go to a huge excess of quencher, all of your photons may be absorbed by the quencher, not by the molecule, and they'll basically kill your photoluminescence that way. So you have to be aware of the possibility of the quencher absorbing at the excitation wavelength and correct for it when necessary. When you have a phosphorescent compound that's undergoing quenching, anything that involves a triplet state, you need to make sure that you don't also accidentally introduce oxygen to the sample, because that itself will quench phosphorescence, as we also talked about last time. So you want to make sure that whatever quenching you observe using this Stern-Volmer relationship is only occurring because of the quencher that you're intentionally introducing. Now, sometimes we do measure quenching involving O2, so in that case, of course, you would introduce O2 as the quencher. But if you're doing it with a separate quencher molecule, something different than O2, you have to make sure that O2 doesn't accidentally find its way in also, so you have to have good air-free techniques for that. You want to ensure that there's no irreversible decomposition. So you want to make sure, again, that the quenching process is a sort of bimolecular electron or energy transfer, not that you're just reacting the two things with each other irreversibly and reducing the amount of photoluminescent molecule, and that's why you see a reduction in intensity. And then finally, as you do these types of experiments, you have to determine the appropriate quencher concentrations to use such that you will see a reasonably large attenuation of the quantum yield in the lifetime that you can measure. And that's often done by trial and error. You'll just add in a little bit of quencher at a time. Depending on the inherent KQ, depending on the lifetime, you'll see different amounts of quenching at different concentration ranges. And so you'll adjust the concentration of the quencher until you see a significant amount of quenching occurring of both the quantum yield and the lifetime to be able to measure it out and determine the correct linear trend using Stern-Volmer analysis. So those are all things you'll have to do when you run the experiments. Not a lot of detail there, but that's some things to keep in mind. All right, the last 3 slides here are going to go through some simulated Stern-Volmer data and 3 possible scenarios that you can come across when you do a Stern-Volmer measurement. So another valuable thing that Stern-Volmer data does for you is it tells you the type of quenching that's occurring, which we haven't really discussed yet in these terms. But one possibility is dynamic quenching. This is the type that we've kind of framed all of our arguments in so far. So dynamic quenching occurs when you have a bimolecular reaction of your excited state with a quencher. So you excite the molecule, and while it's excited, it diffuses to the quencher, and those two react with each other via energy or electron transfer. So this is kind of what it looks like. It can be energy transfer or electron transfer, as we said, but the key is that you excite the molecule first, and then it diffuses to whatever quencher or acceptor molecule is present that it's going to react with. And so in that case, when you have dynamic quenching, your Stern-Volmer plot is going to have that linear appearance, both in terms of the intensity and the lifetime attenuation. So when you measure the quenching of the photoluminescence in terms of its quantum yield or in terms of its lifetime, you'll get the exact same Stern-Volmer relationship, the exact same slope, KSV, and the 2 plots will overlay perfectly with each other. This is again simulated data, but we have real data from our lab that looks just about this good, where the 2 plots will overlay perfectly with each other if dynamic quenching is occurring. And so in that case, whether you measure it in terms of the quantum yield or you measure it in terms of the lifetime, you'll get the same type of plot, the same slope. All the parameters will come out identical to each other. So it is valuable to do both the intensity-based and the lifetime-based quenching analysis because, again, those two together can tell you what type of quenching you have. And this is the first possibility where the 2 overlay, and that tells you have dynamic quenching. The opposite extreme is what's called static quenching. We haven't really introduced this yet, but static quenching involves a ground state association of the donor and the acceptor molecule. And so it's not that you're exciting the donor and then it's diffusing to the acceptor. The 2 species are interacting with each other in some way in the ground state. So here's what this sort of looks like. You have your donor and acceptor, again, not excited at this point, and they have some sort of ground state interaction. That can be, a lot of different aggregation processes. It can be hydrogen bonding, it can be pi stacking, it can be a coordination interaction, but something that brings the 2 together and associates them with each other in the ground state. So the molecules of D that are free, the ones that are unassociated, they undergo luminescence as they normally would. But once they associate with each other, the donor and the acceptor, when you excite the associated pairs, you don't see any photoluminescence at all. So basically this whole thing is completely quenched. So the result of this is you're going to reduce the intensity of your photoluminescence because you're taking molecules of D that are, if that's your luminescent molecule, you're associating them with your acceptor, and you're reducing the amount of donor molecules that are available for photoluminescence. So that reduces the intensity. However, the only luminescence you're observing is from the free donor. You don't see any luminescence from the associated pair. And so because you're only seeing luminescence from the donor, its lifetime won't change. Again, the free donor doesn't interact with the acceptor at all in the excited state. And so you're going to basically just see the lifetime staying the same. So that's kind of what happens then with the Stern-Volmer plots. You'll see that normal linear relationship in terms of intensity, but the Stern-Volmer plot is going to be static for a lifetime. Basically, there's no change in the lifetime when you have static quenching. All right, so here's what those would look like mathematically. The quantum yield or intensity-based relationship is going to follow the normal Stern-Volmer relationship, linear relationship that's shown in black here, and this plot down at the bottom. but then the lifetime will be constant. Basically, the lifetime won't change, and so that ratio will stay at 1. So that's this red trace here. So if you see this situation where you're seeing a reduction in intensity but no change in lifetime, that would cue you into static quenching, which means, again, you have a ground state association that's resulting in the reduction of the luminescence intensity. So that's the opposite extreme, static quenching. And then the last possibility that I'll talk about here is where you have both of them occurring in the same process. So basically, the donor and the acceptor can associate in the ground state, and that quenches at some level, but then there's also can be a dynamic component where if you excite the donor, it can also diffuse to the acceptor and quench following excitation. So in this situation, for lifetime Stern-Volmer plot, the lifetime-based Stern-Volmer plot, again, you're only going to report on the dynamic quenching when you're measuring the lifetime. So you will see the normal linear relationship, and you will be able to extract a Stern-Volmer constant that is associated specifically with the dynamic quenching component. All right, and so that's going to be the lifetime-based one, and then for the intensity-based Stern-Volmer plot, it's going to look quadratic because you're going to have both dynamic and static quenching occurring, and the overall quenching is going to end up being the product of those two. May not be mathematically obvious why that's the case, but that's what you'll observe as a quadratic dependence on the quencher for the intensity or steady state-based Stern-Volmer plot. So here's what the data would look like. So the lifetime-based plot, which is shown in red, is going to have the normal linear appearance, and by fitting this line, you can get the Stern-Volmer constant here for the dynamic quenching. And then what you would do, it's not going to be very reliable to try to fit the quadratic data and extract two floating terms here, but you can fit the lifetime-based approach, the lifetime-based data to get your dynamic quenching constant. You fix that value, and then you fit the black trace here, floating the other Stern-Volmer constant for static quenching to get that value as well. So again, there's value in doing both the intensity and the lifetime-based quenching experiments because the two of these together allow you to very reliably extract both of the Stern-Volmer constants. So these types of complicated situations with both static and dynamic haven't really come up in our own research that I can think of, but it is a possibility, and if you see curvature in your Stern-Volmer plot when you're measuring it in terms of intensity, that is likely the reason that's occurring. All right, so hopefully that's helpful for those of you that missed the workshop or wanted to go back through some things. So again, I apologize for the technical issues. This is not the exact video that I recorded, but that should give you an idea of what we went through today. I'll send out the slides as well, and we'll be back next week with hopefully full technical capabilities and a discussion of different classes of photoluminescent compounds. All right, thanks for participating, and I'll see you guys at the next one.